So yes, fungi are just amazing organisms, and I'm hoping to show you some of the wonderful connections they have in our ecosystems and tell you a few stories. And I do like questions as we go, because if you wait until the end, I'm probably not going to remember what tangent I was running down when you thought of the question. So, you know, wave your hand, stand up, get my attention, and we'll try and weave it into the story. So if you want to press down, we'll go to the next slide. So, in case people aren't aware, sadly, dictionaries haven't kept up with biology. So most people, if they read a dictionary and said, what is biodiversity? It'll say the diversity of plants and animals. And fungi are not plants and they're not animals. They're more closely related to animals, but that's not very related. So there's a bunch of other kingdoms that we haven't necessarily heard of. And the true fungi are in a kingdom all of their own, called the Eumycota. So if you want to sort of try and spot words that have fungus in them, myco is Latin for fungus. So anything that's got myco in it probably has something to do with fungi. And also through my talk, if you notice, orange texts will usually indicate that fungi are involved somehow. So I've also made orange text for the Chromista and the Protozoan. And that's because there are groups that used to be thought to be fungi that actually, now that we know the true lineages, they're not fungi anymore, but they're still studied by mycologists because we're usually a fairly inclusive bunch, and just because it's no longer in the same kingdom doesn't mean that it's not worthy of attention. So yeah, fungi are in their own kingdom, and then we've got fungoid or fungi-like organisms. Can we have the next slide, please? So, fungi are different. They're really different, and we aren't very good at thinking about them yet, because we're discrete organisms. I can't walk through you, you can't walk through me. So we kind of have this kind of contained little units moving around. That's what life is. But fungi are different. Some people think the mushroom that pops up in their garden is the whole fungus. It's not. It's just the flower or the fruit. A bit like if we only saw apple trees when they were flowering and fruiting, it would be kind of strange, but that's what fungi are. We're just seeing the fruit when the body has got enough nutrients and it's time to reproduce. So what is the body of the fungus? Well, it's called mycelium when it's the collective, or individual threads are called hyphae. And basically these are the most wonderful threads that go through things. They're through leaves, through logs, through the soil. And if we can just press down once. And the great thing about being threads is you can actually live with and past each other. So the same way we can have multicolored spaghetti in a bowl and, you know, pull out individual yellow threads or red threads, they can all still be in the same bowl and in harmony. So fungi can actually literally live, different species, very unrelated fungi can live in the same space and pass on information. They can actually communicate with each other, and sometimes they do argue, and then they actually will put out little chemicals saying, this is my food, go away. So speaking of food, how do fungi digest? They're threads. I've got a stomach. I make great habitat for my microorganisms that help me feed, and I know that I'm utterly dependent on them, but I've made this perfect little pouch, this great habitat for them. What do fungi do? They don't have a stomach. These are just threads in things. Well, they are weird. We ingest our food and then we digest it with the help of our microorganisms, but fungi do it the other way around. They find something, the chemical signals go, oh, this is something delicious, and they actually exude enzymes out of their threads into the soil or log or whatever, and start to break that down into bits that actually go, I want this nice tasty molecule and this nice tasty molecule. So they actually digest and then ingest the bits they need, which is one of the reasons in ecology that fungi are actually quite leaky. They're great sources of nutrients, because basically if you're going to digest your food out there, other things can get in on the act. And so that is why also that fungi are an amazing source of antibiotics, because fungi and bacteria have been competing for the same food. The fungus starts to digest something delicious, and the bacteria, which are very small, go, oh, that's yummy, and they start to breed, and so it's competing. So the fungus, instead of trying to kill the bacteria, what they do is they release antibiotics that just stop the bacteria dividing. And so that's why fungi are an amazing source of antibiotics. So yeah, fungi digestion to antibiotics, all in the one idea, but it's all because fungi are different. They live as mycelium, they're threads, they can all live together. And it's a lot for us to kind of get our heads around. So that's what I'm hoping tonight, is that you'll come away with a few stories of how fungi work and hopefully think just a little bit differently. So next slide, please. So, I've gone into this. I hadn't realised I've kept this slide. So yeah, the summary of fungal digestion. You can read the technical term, but really it's slobber and then suck up the little bits of slobber. And that is what the fungus makes its body with. So if we can just push down one more time. Nope, there's stuff going on at the front. So I'm just going to reiterate this a little bit further and tell you a story deep in the soil. So the soil is an amazing place. There's all sorts of things going on in soil. And to give you some idea of scale, these teeny tiny rods are bacteria, which are, 2 or 3 microns big, which is 1,000th of a metre. And this is a nematode. So nematodes are worm-like creatures that go through the soil. They eat bacteria, they eat protozoans, they eat all sorts of things. So they're quite a motile organism. And these threads here are the hyphae we've been talking about. And you may notice there's a loop there. And if you notice there, the fungus, this poor nematode has gone through that loop and got trapped by the fungus. This is a nematode trapping fungus. That's what its job is. And it's actually used in agriculture where eelworms are a problem, which is nematodes forming clubs on roots, reducing their nutrition. And you see how this is fuzzy. This is not necessarily fuzzy because of bad photography, but it's likely that that is where the fungus has started to exude enzymes and digest this poor nematode that is now fungus lunch. Next one, please. So, I'm now going to lead you through my ideal ecosystem and point out where fungi are hanging out. And if you do want a good, healthy, self-sustaining ecosystem, there's 2 things you need to begin with. And one is a really great planet, and we've done really well there. And we need a nice, warm sun to charge the system, and we've also done well there. So now I'm going to lead us on into our olive leaf here and talk to you about photobionts. Now, I'm going to throw some big words at you, and I'm going to explain what they mean, and it's not because I just like throwing around big words, but what I know is if you have a new word, even if you don't remember that word, but you remember the story, you actually have new space in your brain. And when you have new space in your brain and new ideas, you actually think differently. It's how our neurons work. So by giving you a word and an idea and a story, it will actually start to shift how you think. And I'm hoping you will all start to think a little bit about the fungi and what they're doing in your ecosystem. So photo is simply light, and bios is living thing. So these are the living things that use light, which are plants. They photosynthesize. They take carbon dioxide from the atmosphere, they use energy from light, and they turn it into sugars, and that's what they build their bodies out of. And this is these are called the primary producers. So beautiful trees, orchids, we've got bryophytes, so mosses and liverworts, and algae. And I've got a picture of lichens in the middle here. And lichens, so even in the plants, not true plants, lichens photosynthesize. So if we can go down one more. Lichens are actually a symbiosis. So they are living things living together, mostly for the benefit of both of them. And one of the exciting things that we've discovered in the last 5 years about lichens is that it is actually often a couple of fungal partners making a body for their photosynthetic partners, which are algae or cyanobacteria. And so until about 5 years ago, we thought it was just one fungus making the house, and now they've actually done DNA work, and there's actually sometimes another fungus hidden in there. And sometimes the interesting novel chemicals in lichens is because of that second fungal partner. Now, I'm not on first name terms with lichens yet. There's a lot of diversity out there. I'm still getting my head around it, but I do notice lichens. So I invite you, when you see a lichen, just look and go and wonder and say, are you a crustose lichen? Are they flat and lying in their substrate? Are you a fruticose or shrubby lichen? Are they standing up? Or are they leafy lichens? And I promise you, if you just pause and wonder what kind of lichen it is, just even its shape form, you will start to notice lichens popping up everywhere. Can we go down one more, please? So, as an ecologist, I'm interested in nutrient cycles. So I've told you that fungi are leaky with their nutrients. The same is true of lichens. And I've told you that in photosynthesis, we capture carbon dioxide, so we've got a carbohydrate source from plants. But the other thing plants and animals like us need is proteins. And for our proteins, we use nitrogen. It's a carbon backbone, but there's a lot of nitrogen in protein. And nitrogen is actually limiting in the environment. So it's one of the things that drives ecosystems is the available nitrogen. And so I'm now going to tell you that cyanolichens, or lichens that have a cyanobacteria partner, are a source of nitrogen, which is really great. So cyanobacteria actually can take atmospheric nitrogen, which is in a form that plants can't use, and they turn it into a form that they can use and their fungus partner can use. And it is the same form that plants can use, and it's water soluble. So when it rains or when there's mist or fog, that water passing through those cyanolichens actually dissolves some of that nitrogen and passes down. So there are cloud forests in South America where they've calculated how much nitrogen is being given to the environment. We're talking cloud forests here, so big forests with Spanish moss dripping, lots of orchids and ferns up in that canopy, but all of that thick lichen cover in the branches. 30% of that ecosystem nitrogen is coming from the cyanolichens. And any group that is contributing, at least sometimes, 30% of an important resource like nitrogen, I think is doing something really valuable. So I used to get phone calls, I was the fungi lady, and people would ring me up and they'd say, how do I get rid of the lichens from my fruit trees? I'm sure they're doing harm, they're terribly unsightly. And I'd be like, I don't think so, maybe you should, their habitat. And people weren't really buying that. But if I now tell them they're getting a free feed of nitrogen for their plants, they're much happier about not trying to get them off their trees. So there we go, we've wandered down the olive branch, and now we're going to wander over to our little lamb, because this is ecosystem connections. So most of you have an idea about herbivores. They're animals that eat plants for their nutrition, and they're a very important group. And I've put up some, some pretty charismatics, but I've also put invertebrates. And I've also already mentioned that gut microbes, so protozoa, fungi, bacteria, all sorts of organisms are really important for organisms like us and these that have stomachs and, do the digestion. I've put orange here, and I've put anaerobic ruminant fungi, and I've also called them chytrids because their old name in the books is chytrids. But unfortunately, chytrid is the same word that is used to describe the fungi that are causing problems for amphibians. So our frogs and salamanders have chytrid disease. These are not the same chytrids, so that's why I'm now trying to use anaerobic rumen fungi. And rumen fungi is exactly where they're hanging out. They hang out in the sheep and the alpacas and the buffalo. So one of the reasons these ruminants have 4 stomachs sometimes is to make the perfect, without oxygen, which is what anaerobic means, habitat. Because grass is actually really not very nutritious. And even with a whole lot of chewing cud, you are not going to break up the nutrients very easily so that your poor stomach can digest it. But there is a very old lineage of fungi that evolved before we had our oxygen atmosphere, and they have disappeared into the stomachs of these organisms, and they are what does the digestion. They break up those bits of grass that have been beautifully chewed, and that they kind of break it up so then the cow's stomach can actually absorb the nutrients. So most people have no idea that a lot of the animals on which we, I'm wearing wool, I might eat lamb occasionally, we're dependent on these animals, and they are dependent on a whole group of fungi that we still know very little about. Next slide, please.
So, with regards to the rumen fungi come from? Are they on the grass or
They're probably passed with the colostrum. And yeah, we still don't really know, but yes, very sterilely raised sheep or young don't have very good rumen fungi. So yeah, some of it might also come from the habitat, but there's so much we don't know about how things happen. We know when it works, a well-managed herd, good healthy grass, but yeah, when drenching becomes too much or they're raised with, not enough connection to nature or their parents, they have digestion problems, and probably it's an absence or a low diversity. One of the things we're really discovering about stomachs and digestion is that a diversity of organisms makes for a stronger digestion, and that's because different organisms are better at getting out different bits of nutrients. But for the anaerobic rumen fungi, yeah, that is a really big group, and ruminants are really dependent on them. And yeah, I never heard about them when I was doing anything agricultural. Next one, please. So I've kind of skipped over omnivores because I think you probably know what they are, animals that sometimes eat plants and sometimes eat other animals. So now we're going to move to the carnivores, and they're a sexy bunch and they're very important for ecosystem management. But I have popped my nifty nematode trapping fungi slide here. I've also put up a sundew, just to remind you that things that eat other things aren't always sexy, fast predators. So sometimes they just sit and trap. The nematode trapping fungi in soil, and the sundews, basically, which can't flower unless they've eaten a couple of insects because they don't get enough nitrogen. That's why they're trapping invertebrates, is to get that little bump so they have the energy to flower. Next slide, please. So that's ecology that most people get taught. They get taught these quite simple food chains about plants that get eaten by animals and animals that eat other animals. But there is a bit more to our ecosystems. So now I'm going to take us to our praying mantis icon and remind you about decomposition or recycling. We are a resource. We're a resource when we live for our gut microbes, and when we die, we are a bunch of resources. We're nitrogen and carbohydrate, and so are plants. So logs and leaves and litter are also resource for other life. And it's an endless cycle, and without decomposition, life wouldn't actually be doing as well. This recycling of nutrients is really valuable. And I have put up these pictures of all these invertebrates because invertebrates, the largest group of the animal group, is doing a really important job. Most of you if you've ever made compost, you will know if you made compost with big chunks, it goes really slowly. It doesn't actually become useful to put on your plants quickly. So you chop it up and you make it nice and fine, and then you've got good compost faster to then help your roses or your orchard or whatever you're trying to make grow. Nature's chopper-uppers are invertebrates. Worms and protozoans and these micro-invertebrates and even beetles. They are the things that do all the chopping up. And because we kind of often have quite a simple thing, we think these guys are eating wood, particularly beetles. People go, oh yeah, borers, the larvae of our beetles, they're eating wood, and we think that's what they do. Wood, the lignin in wood, the stuff that makes wood hard, is a really tough chemical. It's worse than plastic. It's really, really hard to break down. And there is one group of organisms that basically have the right chemical set in their enzymes to break it down, and they are the fungi. So they are the wood-rotting fungi, and without them, all of that woody material would stay as wood, which is great if it's a table or a lovely lectern, but not so great in an ecosystem where you want that nutrient released for the next generation. So all of these micro things that eat wood are digesting fungal hyphae. They can't break down the wood. They are not termites. They don't have protozoans in their stomach that can kind of break down the wood. And even termites can't break down the lignin part. They're only breaking down the cellulose part. So it's all those strands of fungal hyphae in the log or in the tree or in the table that are breaking down the wood and releasing those nutrients and helping with that ecosystem cycling. So it is utterly critical that we have these decomposition cycles. and we need everything to be working together to get recycling going so that we have healthy ecosystems that have enough nutrient. Yep. So humans have kind of known that pre-digested food or fermentation is actually easier to digest. We have been using yeasts and molds, which are true fungi, but they tend to have less mycelium. They tend to be more, a bit like bacteria. There's a whole lot of budding going on. So split, split, split. And without these guys, we actually wouldn't be getting as much out of our food. I'll tell you, raw chocolate that actually hasn't been fermented or pre-digested by fungi doesn't give you the dopamine precursors. You don't get the good chocolate rush for your brain because those chemicals are still locked up and too long for you to be able to absorb. So the fermentation stage, which is actually in the drying stage of cocoa, is critical. And the fungi are there, they're wild yeasts. And so, and most cocoa still comes from developing nations where there's, it's not as sterile and it's not as controlled. And so we get great tasting chocolate. I also am just a fan of all of these. If I only had these, I would survive. So humans have been using fungi for a long time. And it is that basically pre-digestion or fermentation, breaking down big long chemicals into smaller chemicals that we can use. Next one, please. So this is one of my concerns because I don't think we've understood ecology very well yet. And in the 20 years that I've been studying mycology, there has been a lot of energy, and rightly so, put into our birds. They've been canaries, they've been singing their hearts out that there is something wrong in Australia's ecosystems, and the bird populations have been dropping, and we've been trying to help them. My problem is we've spent a lot of time on birds, and I'm not sure that we've been always very successful. I think some of the connection corridors have done some good, but there is still population drops. And in the last couple of years, we now know that there is an invertebrate population drop globally. We are losing the invertebrates, which worries me. Nutrient cycles are slowing down. And at the beginning of that nutrient cycle are the fungal hyphae and the bacteria and the protozoa. Why are they slowing down? Is there less growth? Are there less fungi? We don't know. But if there is less food for these guys, there is less food for our birds. So we need to get clever, and we need to save our birds, we need to improve our habitat as we can, but we need to understand the underlying ecosystem, and I think fungi and how they live and what they need to live is going to be part of this solution. So I'm hoping that we use the most amazing technology that we now have to really start to answer some of these questions so that we can save our birds before it's too late and have good, healthy ecosystems that do have good, healthy nutrient cycles. So decomposers are utterly critical. We need them, and we need them to be working. One more. So this is a tree that was near me in Melbourne that sadly failed its being a tree and allowed to stay job. And a lot of people think fungi, particularly bracket fungi in trees, is bad. And this tree, I can understand, probably did need to go. You can see there's 2 footpaths, and I'm actually taking a photo on the 3rd footpath. So yes, trees with disease in them on footpaths probably do need to go. They probably are a risk, and probably the council arborist was correct to take this away. But we shouldn't clean up all of the time. We actually need some pathogens. If we can go down one more. So yeah, this is the bracket fungus that probably was the death knell to this lovely eucalypt. And they would have gone, oh, it's in the heartwood, that tree's doomed. Good pathogens, good diseases don't kill their host. And there are bracket fungi in this country and other countries that are literally 1,000 years old. So they have been living on, they are they're pathogenic, they're living on a living tree for 1,000 years. They're a good pathogen, they don't kill their host. And those, often that branch that gets a wound that pathogen gets into, that's the first step of decomposition. that pathogen starts to decompose that wood. Other wood rot fungi get in, a beetle will crawl in and start to eat that fungi and makes a hole. And when you have a hole in a branch, you've got habitat for something to live. You've got a hollow. Hollows do not spontaneously occur. They are part of the ecosystem processes, and unless we wish to be running around putting up nest boxes that need to be replaced every 20 years or so because they fall out of the trees, we need these processes. So utterly understand that the council took this tree out, but if it was in the middle of a paddock where nobody walked very often and it didn't matter if it dropped a branch occasionally, we need to keep those trees, they're habitat trees, and we need the fungi in them doing their job, which is ticking away, taking a bit of extra nutrient, making hollows. We need all of the fungi, even the bad ones. Yep. So I'm just going to tell you a little bit about a weird group of fungi and a relationship they have with plants. So these are plants that actually live on fungi that live on decomposition. So they've got the most amazing name. It's myco, meaning fungus, hetero, which means different or diversity, trophic is how you get your food. So these are plants that are living on fungi that are breaking stuff down. Fungi are called heterotrophic. The nutrient recyclers are heterotrophic. And Australia is actually a centre of diversity for some of these. So some of our thismias and our orchids are actually living on fungi that are breaking stuff down. So some of those large logs that are being turned over, and I know that there are, up here in the Adelaide Hills, there are orchids that are basically living on the fungi that are breaking down the logs. So those big old logs that sit there for decades, slowly decomposing, are habitat for the fungi, and the orchids basically live on them and steal a bit of their nutrients and put up the most beautiful flowers. So an amazing group and kind of the weirdest, lifestyle that I know of yet. I'm sure there's probably something weirder, but it's pretty weird. And yeah, I love that Australia is a centre of diversity for them. It means that we've had these nutrient cycles going on for long periods of time. Next one, please. So that's been our little mantis moment. And you'll probably have realised that we still have mushrooms to get to. What more could this woman possibly say about fungi? So I'm going to throw a new word at you. Bios, Greek, living thing. Troph is how you get your food. So these are living things that get their food from other living things. And it's a bit of a continuum. Sometimes it's symbiosis and all good and they both get something out of it. And sometimes it's not so good and one partner gets stuff and the other partner doesn't do so well. So, yep, there's some bad guys. We've got some galls and rusts and smuts. And I will talk to those first, but there's also a bunch of good guys that I'm going to lead you through. So, a lot of people will have probably a much better handle on some of the fungal diseases if they have any sort of farming background. We have been at war with these guys for quite a while. And unfortunately, in the 20th century, our thinking got a little bit fuzzy. We thought, if we love this food, let's make it easy, we'll mechanise, we'll grow all of this food, same gene, same low diversity in the same place for very large areas. But unfortunately, when we do that, it's not only our favourite food, it's a bunch of fungi's favourite food. And pathogens generally take hold as a population control. Something's out of balance, there's suddenly hundreds of this thing where there used to be 4 or 5 in a scattered area. Something will take it down. And the more we try and grow food en masse, the more we're at war with the fungi that are trying to eat our food that we're growing en masse. And I understand that it's probably not going to be this, pretty permaculture, utopia. But we do need to look back to nature, and whether we've got garden rooms or something, we need to stop these massive fungal diseases doing harm, and the reason they're doing harm is because of the way we're growing our food. So I think we can get clever and look a little bit back to nature and stop fighting quite so hard against the pathogens. So that's the bad guys, and I think people know more about that than any other group of fungi. So I'm going to leave it there, had my little soapbox moment, and I'm going to move and tell you about some good guys. So these are a crazy group of fungi. They are called endophytes, inside plant. And this slide up here, which is the background as well, is basically a slide of a piece of plant tissue, which is pale blue, and the dark blue threads are the fungal hyphae. So once again, we've got fungal hyphae or mycelium inside plants. And the people who used to study plant tissue, you know, not everybody gets out a microscope and starts staining up the fungi in it and look at it. It's not normal behaviour. So it was plant pathologists that did this. They were plant pathologists. They were used to looking at plants and looking for disease. So they knew, plant pathologists knew for at least 100 years, if they took any healthy plant from a healthy system and looked in their tissue and dyed it up, they would find these fungal threads all through the plant, in the flowers, in the leaves, in the bark, all through the plant. And so they invented a term. They called it latent pathogens, because they were plant pathologists. They knew that at some point this fungus was going to turn and there was going to be a pustule or a necrotic bit of tissue, and they were going to see the evidence of the harm this fungus was doing to their plant. But they couldn't see it yet, so they had to call it a latent pathogen because they couldn't see the evidence of the harm that they knew was going to happen. Luckily, there were some good scientists amongst these plant pathogens, and they kind of scratched their heads and kept looking for this harm. And so the idea of endophytes came in. Maybe some of these fungi aren't doing harm. Maybe they're not all bad. And DNA technology really helped. When DNA was first started to be being done, some of it in the 90s, I had this bamboo. I was at a conference and this guy came up and he was a bamboo expert, and he'd been investigating his bamboo and was trying to get DNA out of it so he understood his bamboo better. And he said, "Your fungi," when I said I was a mycologist, "I keep doing samples and all I get is fungal DNA." And I was like, cool, wonder what the fungi's doing in all of that bamboo. And now I actually know that bamboo has the most amazing series of endophytes in it, which are fungi that are helping. They give disease resistance. So if a plant is sitting out there and a rust spore, so one of these terrible plant pathogens, lands on its leaf and starts to tap down and eat that leaf, the endophyte already in there will actually start to fight that rust spore and say, my plant, my leaf, go away. And so they actually act as a bit of a defense mechanism for the plant. They also, some people may know that when you have an area and grazers come in and they start to chew on the plants actually start to taste bad. The grazers don't like it after a day or two. And that's because basically the plants start to produce things that make it taste bad, tannins and other alkaloid chemicals. Sometimes the plants aren't actually making the things that taste bad. It's actually the fungal endophytes making them taste bad. And so they, move on the herd, unless of course you put fences up and they can't get out. And one thing that we're starting to really know about endophytes is good healthy plants with lots of endophytes actually more drought resistant. They've grown tomatoes with endophytes and tomatoes without endophytes, and the ones without endophytes basically need twice as much water. So why wouldn't we want good healthy endophytic populations in our plants? They also might help just store carbon, which can't be a bad thing today, can it? So endophytes are cool. And mycorrhizae, probably if mycorrhizae had been found second, we'd just call these root endophytes, but mycorrhizae were actually found first. So the scientists looked at the roots first. Myco, fungus, rhiza, root. And this is where the fungi hang out. And you see they're shaking hands. This is a good relationship. They both get something out of it. So if we look up here in the corner, we've got a teeny tiny pine seedling, very small. Lots of people have probably planted things and they've planted them and they just sit and they sit and they sit. What on earth are these plants doing? What they're doing is they're producing a large area. Their root mass has got to get out. So the kind of spaghetti under the pine seedling is the large root area this tiny pine seedling is going to need to get going. And it is covered in white fuzz. And you can see an even larger area of mycelium. So this pine has partnering, even at only half a dozen leaves up the top there, It is partnering with fungi, and those fungi are going further into the soil. They've got great enzymes. They can get the phosphorus or the nitrogen or whatever the micronutrient is, boron, and they pass it back up to their plant. Their plant is photosynthesizing and passing down carbohydrate. It's a really great system, and when plants started to scramble out of the creeks way back when, you know, whatever it is, a billion years ago, there were fungi around, and there are fossil evidence that basically fungi and plant have had this relationship right from the beginning. So 90% of terrestrial plants have fungal partners. Next one. Yep.
I've sort of read about mycorrhizal fungi and you know, asked somebody like, how do you know you got it? And he says, well, you should be able to see it. Well, I sort of read that it's too fine for you to actually see with the eye.
Yeah.
And so they said, well, compare it with a plant of dust. I said, well, how do I get a plant of dust if I don't know where it is?
Yes. So yeah, it is.
Looking at increasing my veggie art sort of thing.
Yeah, if we can maybe just go back one. It is really hard. So you can see this fuzz, and I'm just trying to see if I've got a picture of it. I don't actually have a picture of it. Some roots will actually, if you grow them with mycorrhizae and without mycorrhizae, the root form changes. So usually if you've got roots that have actually got little, in the pine family anyway, double bends, that is actually shows that they're mycorrhizal. But yeah, this is not my super area of expertise, so we really want a mycorrhizae expert, but I do know that you can, the way the root grows with mycorrhizae is different to when it grows without mycorrhizae. And certainly non-mycorrhizal plants have just the most amazing roots, because basically they don't have fungi partners, so those roots have to do it all. So if you ever look at proteoid roots or from the Protea family, they have the most amazing, super branched, crazy root. You just kind of look at it and go, oh my God, it's a feather. No, it's a root. What's going on here? And those plants have very, very complicated root systems because they've got to get all the nutrients themselves. But it also makes them very susceptible to root diseases, because basically they've got too many big holes that the diseases can get in. Yep.
Just really quickly, so are you saying that mycorrhizal fungi are endophytes?
Well, basically they're biotrophs. So they have the same life cycle, and probably if people had realised that basically there were fungi all through plant tissue that were living in partnership, mycorrhizae probably wouldn't have got a different name. But now we kind of, people actually talk about root endophytes, but they're not mycorrhizae because they're not actually doing quite the same job. We invent these names for these terms to help us understand the organisms. But the organisms don't read the rule book and hear that we've put them in this box and this box. And some things do this and this and this. And sometimes they do it at different times in their life. And we then get very confused.
Because I guess I didn't picture mycorrhizae as something that lived inside the root.
Yes.
So much as in association.
So yeah, there's different kinds of mycorrhizae. So some are ectomycorrhizae, which are a bit like a sock. So the fungus sock is on the outside and it only just goes into the first couple of cells. Yeah. But the original, or one of the earliest lineage of mycorrhizae, or the Glomeromycota, which have these most amazing, beautiful golden spores, this is actually a slide of that inside a root, and they're endomycorrhizae, so inside root. And they have these, they're called arbuscular mycorrhizae, which is arbol, which is tree, and they have these tree-like structures in the root, so they have a super large area for nutrient exchange. And those are the ones that are really common and really hard to see. So yeah, and that's the thing, we've got all these crazy terms, and some of this is a continuum and some of this we're still just learning. Yep.
Yeah, just on the identifying roots with fungal association, I work with crop plants,
yep.
And generally speaking, for crops, wheat, peas, barley and so forth, if you dig the roots out and gently wash them, if they've got a reasonable amount of mycorrhizal fungi, the dirt will cling to the roots because the fungi is holding it tightly.
Yep.
If you wash them and they come out clean, they don't have much of an association.
There we go. That's really useful.
You have to sacrifice a few plants to find out, but yeah.
Yeah, well, I never put an answer of what to look for outside of a comparison, and I can't make a comparison, you know. Yeah.
How long does
I'm going to have to come down and listen to this question. Sorry, hearing aids, you're too far away. I'll come back.
How long does fungi live?
How long does fungi live? It's a really good question. How long do some fungi live? Some fungi are very short-lived. But let me tell you that the largest organism on this planet is a fungus. It's one fungus, and it is something like 1,056 hectares for one individual. They've genetically tested it. It's in an aspen woodland in Oregon, and they've dated the age of that one individual somewhere between 8,000 and 10,000 years. So some fungi are really long-lived, and they are partnering with woodland systems that have been stable and long-lived for a long time. And I just think in Australia we need to get our act together, because I reckon with our river red gums that are so old that I reckon some of those are probably older and probably partnering with the fungi that may be older. So, you know, maybe we can get competitive and see if we can find an even larger, older fungus. So, Mycorrhizae are awesome. And is it gone to sleep? Have we killed it? Oh, the technology. Let's maybe just try and hit escape. And there we go. Okay. So, the simple graphic of the plant, it photosynthesizes, yep, and then down. The fungus is really good at getting nutrients out of the soil. One more. So they partner. It's pretty cool. Yep. Next one. So the even cooler story, and this is something that people have been living in nature and observing for a long time. We've known that somehow plants communicate. When I'd started studying biology, I was a student in the 90s. And at that point, we knew that plants communicated, and they actually reckoned it was this thing called W waves, a bit like Wi-Fi, that somehow a plant would get eaten and distressed, and it would communicate its distress to other plants. And they thought it was, yeah, a bit like radio, a bit like Wi-Fi. They were making it up. They were putting up an idea. They could see the evidence. They weren't sure how it happened. We now actually know it's not Wi-Fi, it's not in the airwaves, it's actually physical connection. So if you go down, basically this plant can get chewed and it will communicate probably through the endophytes down into the mycorrhizae and let the plants either side of it know that it has been chewed, and therefore you need to be aware there's herbivores in the area, and if you can do something about it, maybe you should. So there is actually chemical passing. And also for us as ecologists, we need to know that plants pass sugars to their fungal partners, and sometimes their fungal partners don't need all of that sugar. So this plant here is not doing so well. We're actually going to give a little bit of extra sugar over here. And some plants will actually produce more carbohydrate to feed to their fungi when they're passing it to relatives, plant relatives, so my progeny, and they're called mother trees. They actually help give a little bit of carbohydrate to get their seedlings up. And other plants are less fussy and will actually pop up totally different, non-relative, but in my ecosystem, you know, my community. So the fungi literally are propping stuff up. And this is one of the things that we don't necessarily understand. So when we go, oh, we're going to clear this undergrowth, we're going to get rid of this stuff, we don't know if we're taking out the things that maybe are better at getting nitrogen and actually passing stuff back up in the other direction, or is it summer and things are passing this way or that way? We don't always know. So we don't know what we're messing with sometimes. And I think we need to get a little bit cleverer, because yeah, if the fungi are communicating and passing sugar and this whole network, they're actually holding it up, which is great. It's propping up the ecosystem, but it does mean that when ecosystem collapse happens, the plants are in control. I'm a plant, I have a few dozen relationships at any one time with different fungi, and I keep all of those fungi on tap. I give them a little bit of carbohydrate. When they're doing something particularly good for me, I don't have enough phosphorus. These three fungi are actually really good at getting phosphorus, so they're giving me most of my phosphorus. I give them extra. Or it's a wet year, I haven't got enough oxygen. The ones that are actually better at giving me kind of a bit more air, I give them. You know, the plants are in control. But when a plant gets super stressed and I can't live, I stop feeding all of my fungi. I only feed the fungi at that point that are helping me. And so I've gone from having a few dozen relationships to a few relationships. And then the climate change shifts. And all of those partners I used to be able to get lots of different things from are no longer there. So there is strength in diversity. Mycorrhizae is not one fungus, one plant. It's plants connected by fungi in diversity. And that's what we need. We need strong diversity of the fungi to help healthy ecosystems. And unfortunately, humans have done a really good job of simplifying our environment. So this is a picture of Mark Newbound trying to just give you an idea that when you walk through a grassy woodland, that probably there are lots of different individual fungi and lots of different species. I'm actually the spaghetti bowl of fungi person, so if I did this picture, it would be surreally rainbowy, interconnected, coloured, and you'd probably all fall over and vomit. But I do like it as a graphic to just remind you that when you're walking across the environment, you're passing across lots of different fungi, and we usually only notice when they pop up their little fruiting bodies. Next one, please. So how long have I got? 15 minutes? Okay, I need to stop telling quite so many stories and maybe talk a little bit faster. So, next slide, please. Yeah, move your chair over. So, fungi are food, and they're really important. I've told you the beetle story, so I'm going to skip over that, but I am going to go to my marsupial and just tell you that Australia is the centre of diversity for truffle-like fungi. Truffle-like fungi are mycorrhizal, so partnering with plants. And basically, when Australia started to dry out, if you're a mushroom and you pop up in a drying wind, you actually don't get to spread your spores before you've dried out. So there has been evolutionary pressure to stop popping up. But fungi still need to spread their spores. They can't just sort of grow here and have my spores here. That's not useful. I'm not going to find a good plant partner if I can't move. Plants also, they have a bit of a dispersal mechanism with their seeds, but seeds often don't go terribly far. So you've got two static organisms. How do they get together and meet the right partners? In comes a cute ground-dwelling marsupial that needs lunch. And basically, we have, with our bandicoots and our betongs and our potoroos, and in fact a lot of our macropods will eat some fungi, but our mycophagus, our fungus-eating specialists, are really good at finding these truffle-like fungi, and in their scats basically is the heaven of all sorts of spores. So when these guys move around the environment, they're basically mixing up the fungal diversity and allowing seedlings to have a better chance of finding the right partners for a healthy system. We've unfortunately broken this chain in a lot of places, and that means we've basically got long-lived trees and plants, long-lived fungi, but they haven't been able to mix up and change partners. So there is the potential that this has been a sort of bottleneck 200 years, which is why remnants are so vital. We need to keep that diversity and we need to reconnect that diversity so the fungi can start to move again and help create healthy systems further along. Next one. I'm not going to tell you about fungi and snails because I've told too many stories. Oh, and I'm not going to tell you about fungi on fungi. There's more, there's good stuff. I'm going to tell you good stuff. So yeah, this is our kind of the fungi connections. And really they're the links in the ecosystem, or there's a phrase from a botanist called Raven who called them the ties that bind. And that's it, they're holding the ecosystem together and connecting it. So this is kind of, yeah, I told you that I promised you a big vomitous, brightly coloured mush. Because they're connecting our ecosystem beautifully and doing good jobs. Okay, everybody, you have 10 seconds to stand up and stretch. Just move a little bit. We've got 15 minutes. We're going to get a cup of tea, but some people are nodding off. Just have a... Okay, it was only 10 seconds. Sit down. So I'm going to shift gears a little bit and just tell you a bit more information about fungi generally. So this is biodiversity, diversity of Australia's living things. So the biggest part of the pie is invertebrates. If we add up all the things with a spine, so fish, reptiles, birds, mammals, 2%. If we add up all of the plants, 5%. So I think most of our energy at the moment is spent thinking about 7% of our biodiversity, and I think that's possibly why we're not doing as good a job as we could. So a little bit of data about fungi. They are probably, we now think with estimates, going to be about as diverse as the animal kingdom. So yeah, we've got a lot of fungi out there and not many of them have names. People often think, oh, fungi, they're everywhere all of the time. Not so. In Australia and New Zealand, or Australasia, 72% of the fungi here are not found anywhere else. So if we're not looking after our fungi, nobody else can look after our fungi because most of them are ours. We need to do our job. So just a few ideas about conservation basics. Get to know your local fungi, and I'm delighted to be here for the launch tonight because that is part of that exciting process. And you can't do anything about stuff unless you've got data. We can only change our behaviour when we know what we're doing and what effects it has. And at the moment, we don't know much about our fungi, which is why getting to know your local fungi and monitoring them is actually giving you the power to understand what's going on in your backyard. Do protect remnants, or my fungal arcs. Try not to use fungicides, don't mulch unnecessarily, and stop chopping up nature, it's bad, people. Mycorrhizal fight global warming. Basically, when I was a student, people used to go, oh no, young plantations actually suck up more carbon. And it was an encouragement to sort of chop down old growth forest and put in young plantations. Yes, young forests do suck up a lot of carbon, but it's actually not as much carbon that is held in the mycelium of long undisturbed patches of forest systems. They are holding an amazing amount of carbon. And we want them to keep doing that because unfortunately, with the melting of the tundra, we're about to release a whole lot of methane and other carbon dioxide that we can't do anything about. We can't stop the melting, but we can keep our long undisturbed patches and keep them healthy and hold that carbon in the soil where it should be and not release it into the atmosphere. This is something I really want us to know something about. Understand our spore bank. There's been a lot of work done in the last 2 decades about Millennium Seed Bank and understanding and keeping our seed bank and having a good resource for, revegetation. We don't know which are the good partners. We know that a diversity of fungal partners is probably going to be the best, but we don't know when, where, why, or how. So I want us to know a bit more about this in the next 20 years if we can, and we need to bring back our marsupials if we can. So FungiMap is a national not-for-profit, and we started out as a mapping scheme, and we still do that. And when we started out in 1995, the only people that really had records of fungi were the herbaria. So, a few dozen scientists for 200 years had been making scientific collections, but it's too slow. And at that point, for most species, there was less than 5 records for the whole country. And if you only know where something is in 5 places, you don't know much about that fungus. Next slide, please. So there is power in data. So FungiMap does not make collections. It's observational. We want to see where things are, and we know that you can't learn all fungi straight away. You're not, they're tricky. So we encourage people to start learning the easily recognisable one. Most people don't start learning boring brown birds or wader birds. They're just way too hard. Don't start with brown mushrooms. You're not going to get anywhere. But you can start with beautiful fungi like this, the Amanita. And so after 20 years of gathering data, where there was 5 records of this fungus to begin with, we now have over 2,000 records, and we can really tell something about where this fungus lived. All the purple dots, and then you can start to do modelling. So where the red is where this fungus, with the simple, this is quite a simple rainfall temperature model, suggests this fungus might be. And you can see in South Australia, it's here, but it's at the edge of where it lives. Next slide, please. So I'm probably not going to tell you about the lost fungi because we don't have a heap of time, and I'm much more interested in telling you about a little bit about fungal identification, because that's going to lead us into our book launch. So do start with easily recognisable fungi, and you guys are about to get the booklet to tell you about the local ones here. You can also, if you're interested, interest grows. Fungi Down Under has 100 recognisable fungi for Australia. So these are the ones that are easier to learn. I know they don't necessarily all pop up in your backyard today, but if you flick through the book and get to know them, you will start to go, I've actually seen that one. Or when you do see it, you'll go, this is one I can actually learn. So I do think that recognisable is a useful thing. And so this is Fungi Down Under, and I'm sure, and the booklet we're about to get also has this. Basically, it's some information. It's not one character that tells you what something is. You know, trees, yes, they're tall, they have trunks and leaves. But, you know, you need to know a bit more. What kind of bark type has it got? You know, how big are the leaves? What do they look like? Fungi have a whole set of characters that are like that, and a bit like anything that you learn to identify, there's a bit of effort in the beginning to work out what all these things mean, but they do mean something. Scientists aren't just throwing around long words and asking questions for no reason. And so my suggestion is, before you even start worrying about what species you've got, work about what morpho group you have. And all morpho group means is the shape group. So mushrooms, or have a cap, gills and a stem, is one morpho group. It's the most common macrofungal group, so most of the diversity is in this group because it's a really efficient design. But there are other there's jelly fungi, and there are stinkhorns, and there are club fungi. And so a bit like when you can go, tree, grass, orchid, we can talk about these morpho groups and have narrowed down what it is. And that's the first step to identification, is going, it's in this group, it's not all of those other things. I don't need to worry about those things because it's in this group. And now I can look up this group and have a much better chance of working out what it is.